Welcome to Section 5 of Lab Techniques. In this section, we'll be discussing karyograms and FISH. Let's get started. A karyogram is a lab technique in which metaphase chromosomes are stained and placed in numerical order based upon their size and morphology. This is commonly used to diagnose chromosome abnormalities such as trisomy 21, Turner syndrome, and other genetic disorders. This is also often used in conjunction with fluorescence in situ hybridization, or FISH. I'll explain FISH more in a second. This is a normal karyogram which can be found in Section 5 of Lab Techniques. Notice that the chromosomes are arranged and placed in numerical order based upon size and morphology. So you can see chromosome 1 here, these are very large, then 2, then 3, and so forth. Fluorescence in situ hybridization, or FISH, is a lab technique in which fluorescent DNA or RNA probes bind to a complementary region of interest on a chromosome. The complementary regions on the chromosome can then be visualized with fluorescence microscopy. Just like with the karyogram, this is commonly used to identify gene and chromosomal abnormalities. Let's pull up an image of this so you can visualize what I'm talking about. This is a figure of FISH which can be found in Section 5 of Lab Techniques. Notice that a fluorescent DNA probe, which you can see right here, is combined with a sample of DNA. The probe hybridizes, so undergoes hybridization, with the complementary strand of DNA, which can then be visualized on a chromosome. You can see that right here. This is a figure of FISH which can be found in Section 5 of Lab Techniques. Notice that this is a karyotype of a healthy female. There are 46 chromosomes, including 2 X chromosomes. The green stain is caused by a probe which binds to a specific DNA sequence. The sequence from this picture is a marker for specific chromosome bands rich in genes. The red color represents a counterstain. Let's go through an example to see how this could be used clinically. A 66-year-old male presents with a 2-month history of fatigue, weight loss, night sweats, and several episodes of minor cuts causing excessive bleeding. A CBC reveals elevated white blood cells. Laboratory analysis reveals decreased leukocyte alkaline phosphatase. A genetic abnormality is suspected. What laboratory technique will most likely be used to identify this patient's genetic abnormality? Hopefully from the question stem you notice that this patient has symptoms concerning for cancer, including weight loss and night sweats. A CBC showing elevated white blood cells is concerning for a hematologic malignancy. Finally, decreased leukocyte alkaline phosphatase, or LAP, is a unique finding in chronic myelogenous leukemia, or CML. Perhaps it would be useful if we took a step back and discussed LAP. LAP is an enzyme present in the secondary granules of granulocytes. This enzyme is useful for the purposes of inflammation. Cells that arise to fight infection or inflammation will be LAP positive. However, cells that arise due to CML have no intention of fighting off an infection, so it makes sense that these cells will be LAP negative. Okay, with this in mind, we're asked what lab technique will be useful in identifying the genetic abnormality seen in CML. Recall from pathology that CML results in a translocation of chromosomes 9 and 22. This is also known as the Philadelphia chromosome. As we just discussed, translocations can be identified using FISH, so the correct answer is A. Microarrays are used to analyze gene expression, ELISA is used to detect proteins in the blood, and flow cytometry is used to analyze cell markers, cell size, and cell granularity.